Hello, everyone. My name is Amy Chahotsky of Separation Science, and I'd like to welcome you all to our latest webinar in collaboration with SciEx. Our topic today is challenges and solutions while performing pesticide analysis for the world's largest food and beverage company. Andrew Savage is an expert chemist at Nestle Quality Assurance Center in Dublin, Ohio, where he leads projects for new analyses and technology scouting. His focus has been chemical contaminants in food, and he was previously the supervisor of the Contaminants Laboratory, where he managed the performance of 40 trace-level analytical methods. He has 25 years of experience in analytical laboratories within academia, contract research, and in the food industry, working primarily in mass spectrometry. He holds a Bachelor of Science from Mount St. Joseph University in Cincinnati, Ohio, and a PCQI certification. Angela Simon is a pesticides chemist at Nestle Quality Assurance Center in Dublin, Ohio, where she started in the vitamins department and now works in the contaminants department. She works on most, if not all, of the pesticide methods at NQAC, along with many other methods. She has approximately 18 years of experience in analytical laboratories within environmental, pharmaceutical, and the food industry, primarily working with mass spectrometry. She holds a Bachelor of Science degree from West Liberty University in West Liberty, West Virginia. And so, without further ado, I'll turn the presentation over to Andrew.
Welcome to our presentation on pesticide analysis. My name is Andrew Savage, and I am an expert chemist here at the NQAC laboratory in Dublin, Ohio. Before we get started, I want to thank our scientific partners for allowing us to present and all those who have chosen to attend. We hope that you will learn a little about what we do at the NQAC laboratory and also walk away with some interesting insights. I'll be one of your presenters today alongside my colleague.
Hi, my name is Angela Simon, and I'm a pesticide chemist in the Contaminants Laboratory here at NQAC Dublin. I'm happy to be here to assist in presenting some information about our pesticide testing.
In our presentation today, we hope to give you some insight to our analytical processes for pesticides in food. These processes step beyond the analytical work and are impacted by the workflow and needs of a global company. In addition to the needs of analysis for Nestle, our facility is open to any food producer and offers the same portfolio and results to anyone requesting analysis. We believe that food safety and compliance should be separated from the competitive business landscape, as everyone deserves to have healthy and safe food, regardless of who they purchase it from. In offering these analyses, we have learned some of the best practices and a few watchouts in the analytical process. And it is our hope that you will be able to walk away with some useful information that may improve your knowledge of pesticide analysis and the safety of your food. In our presentation today, we'll go over an overview of the Nestle Laboratory Network, our methodology that we use here at NQAC Dublin, including our instrumentation, the extraction we use, and matrix issues commonly seen. We'll talk a little bit about reference materials and how we use those and handle them. We'll go over our workflow and data handling, which is imperative with all the different types of matrices we get and handling those in a timely basis. A comparison with the recent FDA Pesticide Monitoring Program report and how we evolved to meet client needs in analysis. Starting off, we will go to the Nestle network of laboratories. When we talk about Nestle Labs, we're actually talking about a global network of laboratories. There are 20 NQAC laboratories, which stands for Nestle Quality Assurance Center, across the globe. And we all report into Nestle Research in Lausanne, Switzerland. Our network of lab allows information sharing on method improvements, cross-laboratory validation, and other improvement activities. The Nestle Research Center in Lausanne, Switzerland develops new analytical methods to meet emerging threats. Nestle is active in international standard setting organizations such as ISO and AOAC to work toward scientifically aligned and recognized methods by authorities and industry alike. Again, we feel food safety cannot be considered a competitive advantage. So learnings from our methodology is something that we share with the food safety community. Here at NQAC Dublin, we offer quite a few pesticide methods. We have a large LC pest screen and a GC pest screen. Our polar pesticide method is focused on a handful of compounds and is based off the EU CUPE method. Ethylene oxide was recently added due to customer demand. Our glyphosate method is an AOAC first action method. We also do dithiocarbamates and USP to meet those needs, and soil pesticides. We often get questions about soil pesticides and why a food lab does that. Nestle owns the Libby's pumpkin brand, and long ago somebody figured out that because pumpkin really will pull a lot of pesticides out of the soil, it would be wise to start testing the soil before planting. This helps save a lot of time and money on the pumpkin harvest, as we can screen for suitable places to plant. But of course, we still go ahead and test the finished product as well.
Let's talk about some of our extraction techniques that we use for our pesticide screens. Most all start with a modified catcher's extraction and then proceed to additional cleanups depending on the method. There within lies a struggle to determine which cleanup method is sufficient for the analytes that we are trying to analyze for. Some cleanup methods can react and or degrade the analytes that are being tested for, so careful consideration has to be taken to determine these cleanup steps. For the large LC pesticide screen, the sample extract is split up into two parts at the completion of the catcher's extraction: pre-PSA, which is primary, secondary amine, or post-PSA. As in the description, pre-PSA does not get the PSA cleanup and post-PSA does. PSA can cause degradation to some of the analytes, and therefore these sample extracts have to be divided this way. They are then run on separate instruments. The GC pesticide screen gets additional cleanup using GPC, which is gel permeation chromatograph. It's a type of size exclusion chromatography that separates analytes on the basis of size and organic solvents. This is used for the removal of large fat molecules that could possibly cause interferences with the analytes that we are looking for. Finally, our USP pesticide screen is primarily used for supplements, and these formulations can be very complex, leading to the need for additional cleanup of the sample extract. The sample in this method receives an additional cleanup with carbon black to remove some of the color interferences that could be present. We move on to talk a little bit about our instrumentation that we use for pesticide testing here in the laboratory. For our large LC pesticide screen, in which we screen for approximately 400 analytes, we use an Agilent 1290 paired with a SciEx QTrap 6500 MSMS detector. As mentioned previously when talking about the LC extraction process, our LC pesticide screen samples are extracted and then split up into two parts, one part pre-PSA cleanup and one part post-PSA cleanup. The two parts are run on separate instruments, which allows for parallel processing and also shorter turnaround time. Our pre-PSA cleanup samples are run on an instrument in which the mass spectrometer switches polarity between negative and positive ionization modes. The post-PSA cleanup extracts are run on a separate instrument in which runs only in positive ionization mode. The autosampler is set up to do a dilution prior to the injection for the post-PSA cleanup extract, which helps to eliminate interferences and also keeps the instrument cleaner by doing this dilution. It's very helpful that the autosampler has this capability, as it saves us a step in the sample preparation process. The instrument is equipped with two 54-position sample trays that hold 2 ml vials. We use 2 ml vials for most of our testing here in the contaminants laboratory. This allows us to be consistent across methods and prevents us from having to store additional sizes and types of vials in the laboratory. A C18 reverse column is heated in the column heater compartment and used for separation of analytes. The autosampler is chilled to assist with maintaining stability of the analytes and samples and standards to be analyzed. There is an extensive needle and column wash in between injections and also at the end of each run in order to remove any remaining mobile phase buffer salts that may persist on the column or sample matrix in the needle, which could cause blockage if either are left behind. The runtime per standard or sample is 10 minutes for this method. I think this is incredible that our method analyzed for approximately 400 analytes per injection in this short amount of time. 2 mass transitions are monitored per analyte, allowing us to see the first mass transition to quantitate and the second as a confirmation. This gives us great confidence in our data as it ensures that we are seeing the actual analyte that we are looking for and not an interference that could be generated in some matrices. Our QL for this method is 50 parts per billion for herbs and spices and 10 parts per billion for all other matrix types. We'll talk later in the presentation about how matrix performance can affect our quantitation limits. Next, we'll move on to the GC side, where we'll look at our GC instrumentation that we use for our large GC pesticide screen. We screen for approximately 77 analytes in this analysis. For this screen, we use a Shimadzu Nexus GC 2030 paired with a TQ 8050NX MSMS detector. In our previous extraction discussion, we talked about how the GC extracts must go through extensive cleanup, including going through the GPC for fat removal prior to going onto the instrument. Due to the nature of these matrices, herbs and spices get an extra 3x dilution prior to being injected. These sample matrices can potentially cause havoc and dirty the instrument, which we'll talk about later in the presentation, even after extensive cleanup. 2 ml vials are also used in this method, and we use the direct inject approach. The analytical column is a 15 by 0.25 millimeter by 0.25 micrometer in size, and we also use a GAR column, a short piece of uncoated deactivated fused silica, to assist in protecting the analytical column from contamination, as the dirt injected with the sample will stay mostly at the beginning of the GAR column and will not pass into the analytical column. This helps extend the life of the analytical column, which is a large cost savings. A topaz liner with quartz wool is used in the inlet, which is an inner liner used for trace level analysis. The quartz wool enhances vaporization of heavier analytes and aids in the mixing of the sample with the carrier gas to lead to better reproducibility. It also acts as a type of filter to capture non-volatile matrix components and prevent them from entering the column. We use an autosampler at AOC 6000 for this instrument. Our runtime for this method is approximately 18 minutes, where at least 2 transitions are monitored for each analyte. And as we discussed in the previous slide, when we were talking about the LC-MS instrumentation, the first transition is used for quantitation and the second for confirmation to ensure we are able to identify interferences and that we are giving the customer accurate results. The quantitation limit for all analytes for this method is 10 parts per billion.
Next, we'll move on to matrix issues. When it was first rolled out, Catchers was used for simpler matrix types, such as fruits and vegetables, without processing. The method has evolved with the industry with a significant scope expansion. However, we still experience challenges that are out of the norm. Food producers have a different take on raw materials. Often they look at anything they use at the beginning of their process as a raw material. Because of this, many of the materials are already processed foods, often in a manner that creates challenges for analysis. The concentration or extraction of ingredients that helps with transport and storage tends to increase the material that may cause difficulty in extraction or analysis. Without full knowledge of the concentration factor or what additives have been introduced, accurate and consistent changes to the sampling and sample handling, such as dilutions or slurries, is difficult. Some examples of the matrix types we have seen in the lab are shown below. We've gotten herbal laxatives as a raw ingredient, paprika oleoresin. I'll give you that paprika is within the normal catcher scope, but when you extract the oleoresin, you're starting to push it. And soy lecithin.
Here we have an example of the results in chromatography produced by a similar matrix as discussed above. We are looking at the analyte prothiophos, and the first line of chromatograms represents the second transition of a normal spiked sample matrix. As you can see, two very clear, nice chromatographic peaks at similar retention times. On the line below, we see the first and second transition of prothiophos analyte again, but this time from one of these problematic matrices. This sample is spiked at the same concentration as the sample in the top two chromatograms, but as we can see, there is a large interference that is preventing us from determining the result. Therefore, we would have to report this result as not determinable due to this interference. In continuation of talking about matrix issues, they can affect instrument performance, and they can be impacted by running dirty samples. The dirty source in the picture above is a result of running dirty samples. This can result in decreased sensitivity of your analytes. It can also produce source charging, which is a result of a buildup of compounds that form along the analyte flight path that essentially steal the charge from the analytes that you are looking for and can cause repulsion of your target compound going through the MS, which then can also lead to the decreased sensitivity and linearity. Moving on to unknowns, which also causes significant challenge. Very often we get samples that are submitted with unclear description of what the sample material actually is. For example, the description could say special sample, or the description could just be a number. Often the homogenized portion of the sample gives very little visual clues to what the sample actually consists of. It's very difficult to determine how to perform testing correctly on these types of samples, and we often have to refer back to the customer for an accurate description of what the sample ingredients are prior to testing.
Other items that we've had issues with is reactive chemicals. We have had customers that use food-grade potassium hydroxide as a processing agent in their food, and therefore they look at it as a raw material and they'll send it in. That's not always a great option when you're adding acidified extraction solvent. So that can be an issue. Of course, we always wear PPE and work in a hood. So safety is not a huge concern there, but it does make it almost impossible to analyze the sample. One of the materials we get in quite frequently that can cause significant issues are emulsifiers. Emulsifiers, by their very nature, make liquid-liquid extractions very challenging, as you often get a foam. We've come up with some different ways of dealing with this. Certain salts can help, and also just centrifuging for a little longer can help with that as well. Something that can be a hidden concern or something not so obvious with your controls is overprocessing of the sample before extraction. Homogenization is a critical part of sample analysis and making sure that the results are valid, but overprocessing a sample by blending can heat up the sample, and you can lose compounds that are thermally labile or volatile. So when you have those matrix interferences, what can you do about it? Well, some of the options and solutions that we use are additional cleanup, which is usually the first go-to anytime it's usable. If you have a validated cleanup process that removes interferences, you can use that now and ongoing, and it will help with your overall process flow. One of the things we will use is carbon black. We use that on our USP method, as Angie mentioned earlier, because those supplement samples can really just have a lot of issues in them that create problems for analysis. The problem with carbon black, though, is it's pretty good at removing a lot of compounds, and sometimes your target compounds are lost as well. So we don't use carbon black on our large screen because some of the target compounds get removed by it as well. GPC is super important on high-fat matrices, and we wouldn't be able to run our GC method without it. The GPC removes so much fat from samples that we are even able to do really high-fat matrices such as butter and lard. So it's really a lifesaver for the analysis and for the life of the instrument. Dilution is always a great go-to to help resolve issues with interferences. As Angie noted earlier on the GC method, whenever we get herbs and spices, we automatically do a threefold dilution. This is only possible when you have enough sensitivity. Fortunately, our Shimadzu GC and our SciEx 6500 system offer that sensitivity. So it's something we can go to get a little bit more resolution from those interferences. Whenever doing dilutions or any other cleanups, it's important to have internal standards that indicate what the impact of those additional steps do to your sample and your analytes. Having internal standards throughout the chromatogram can help you correct for any issues that are introduced throughout the extraction. And we use labeled internal standards on some of our more critical analytes just to make sure that they are as accurate as possible. On occasion, we have to use a reduced sample weight when we have a lot of interferences on a sample. We will use a reduced sample weight only after we've tried to run the matrix at the normal weight, because if we reduce the sample weight by our protocol, we have to increase the LOQ accordingly. So this is not usually too big of an issue because we're operating at a low end of the maximum residue limits. And so if we have to raise the LOQ a little bit, we can usually still report a very usable result to the client that has a much better result as far as quality. We often spike for positive control due to the nature of the matrices we get in, especially with unknowns and so on and so forth. Positive control spike will be a spike of all the target analytes at the LOQ. This allows us to have proof positive information that we can see those analytes at the LOQ and ensure we're not missing anything. We still will change our LOQ if that positive control spike does not recover to our expectation, or if we can't see it at all, we may mark that compound as not determinable. This is important to be transparent to the customer as to where the performance gaps may exist on their sample in that analysis. Not a lot of labs do that, but we find it to be very important to make sure that the customer knows what they know for sure and what is still in question. Next, we'll talk a little bit about reference materials that we use for standards and controls. For our pesticide methods, we use custom mixes. It's important to us to have all the compounds reported as a certified reference material to ensure correct identification and quantitation. With this, we monitor 2 transitions for every compound we look for. We run a full calibration curve with every run. Not all labs do this. We do it because, in my experience, mass spectrometers, they can have a mass calibration shifts with even slight temperature changes and other issues. So it's important to have really good calibration data with each batch. The positive and negative controls, talked a little bit about that with the sample spikes at the QL, but we also run a method spike, which is just a blank sample that is spiked throughout the run so that we can see any impact that the reagents have on the extraction. That is very useful if you get a bad lot of material or something like that. It'll tell you any issues you have in the run. Of course, we have a blank as a negative control to ensure that we're not getting any interferences or contamination of the sample. With that, it's really important when you're looking at your overall scheme of controls to know what they do tell you, and probably more importantly, what don't they tell you. Often we talk about the calibration curve is going to tell you about the linearity of the instrument, but it's not going to tell you anything about the extraction efficiency. The sample spike is going to tell you how that compound performed in that sample matrix, but it may or may not alert you to issues you have with your reagents. And that's where that method spike can help us troubleshooting there. By making sure we have overlap of all these controls and duplicates to ensure that we have proper precision, we can make sure that we have quality added in every single run. Some of the challenges, though, with reference materials, especially when you have a custom mix for everything, you have cost and availability. Cost is certainly something any lab is going to take into consideration. So careful planning of how you're going to use those reference materials is critical. Availability it can be an issue, and sometimes you'll have to go to multiple vendors to get what you need for all the compounds to ensure you have full coverage. Several manufacturers now, though, we've found can provide everything we're looking for, and they can do it in the ISO Guide 34 standard, which is important when you're an ISO accredited laboratory. Stability of those standards can be a bit of an issue. Not all these compounds degrade at the same rate, and their expiration dates can differ wildly. So management of all of that is important with the due dates for those expirations, how they're stored. Some of them need to be frozen, some are in the freezer, etc. Monitoring that degradation by doing lot-to-lot checks is important. And then maintaining all the documentation of all this, all the certificates of analysis, the preparation of the standards. It's a lot of work, but that work results in a very traceable, high-quality method. We've often been looking for control materials that we can use to monitor and control chart, and people will ask about p-test materials. And unfortunately, p-test materials in pesticide analysis are not always a great option. Most p-tests are only maybe 40 compounds, and when you have 400, then you're only covering 10%. And we've never seen the use of old p-tests as controls to be reliable because different degradation rates of different compounds. So when we've tried to do this, we've found that you end up spending more time questioning whether or not your p-test is a good control than using it as a control. We've also experimented with round-robin spikes, where we spike a material with a reference material and send it around. But that's also challenging to get a truly homogeneous mixture and to make sure that the degradation issues are not an issue there as well. So keeping good certified reference materials for standards and spiking is a critical part of this quality control. Some of the issues we've had in the past on reference materials and some hard lessons learned are listed here. We had an issue at one point with a custom provider of standards where the carbendazole was mixed in with about 30 other compounds in acetonitrile. And what it took us a few weeks to determine was the carbendazole was only partially soluble in that mixture due to the saturation with the other compounds, and carbendazole is much more soluble in methanol. As a result, though, it was hard to detect because that solubility is very reproducible. So you can inject over and over and over, and you'll keep getting the same amount. But that causes an overestimation in the samples. So the key there is to have an independent control or verification, and as a result, you'll often see carbendazole sold as a single component, even in the mix kits, just to make sure that solubility is not an issue. So anytime you're using a custom mix, having an independent verification that everything is correct is very important. Another issue we encountered at one time that took us a little while to resolve was compounds with the same mass transition. Our company at one time had a policy where the instrument computers were replaced by the standard computers of the company when instrumentation was purchased. And what was not seen at first was a software glitch. It had to do with the BIOS chip timing, where when you scrolled through the different compounds in Analyst, which is what we were using at the time, SciEx Analyst, it showed the same peak, even though it was labeling them differently. So with a mixed standard, that's a very, very difficult thing to see. So going through and making sure that checking this with individual mixes is a way to catch it one compound at a time. And since then, we implement a policy that we maintain the manufacturer computer with all the instrumentation just to make sure nothing like that ever happens again. A big issue that can happen, we saw this several times, especially during the pandemic, is a missing component in a mix. One time we had an incorrect component in a mix. But when you have one compound missing, it creates a huge headache when it comes to making up stock standards again. All your dilution schemes now have to be modified if you want to try to include it back in, or you have to inject it individually. So checking those, again, lot-to-lot checks when you get new stuff in is really important. The incorrect sample, it had a very similar CAS number, and that was something that we caught, mostly because we're using mass spectrometry. The vendor fixed it right away, but again, adds time, so you have to consider this when you're ordering the reference materials. Make sure you have time to evaluate them and correct any issues before the materials you have go out of date. When we have tours through the lab, some people are a little surprised the way we handle our samples, since we're a very large laboratory as part of a large laboratory network. They expect a little more automation, and we certainly do use automation. You can see here our specs genogrinders that we use for homogenizing and shaking the samples. That saves a lot of ergonomic issues, but we certainly keep some manual processes as well. What we have found is that a combination of the two is pretty important to being a robust lab. A manual approach is always going to be a very flexible approach. Sometimes reproducibility, depending on analyst training, can be a bit of a challenge, but that's something we can make more robust with our solid training programs. Automation is pretty great, except a couple issues that we've seen. A lot of automation does linear processing, and that can be a bit of a constraint, whereas a chemistry technician can grab a rack of 18 centrifuge tubes and shake them all at the same time. That can be quite a bit faster. But the biggest issue we would have would be equipment downtime. Equipment downtime that halts our operations is something we have to absolutely avoid, because food is perishable and they need to be able to get their results. So in this case here, you can see we have multiple genogrinders. If one of these goes down, it's not that big of a deal. We can handle that. So we try to build in a lot of redundancy and only automate portions of the process to speed things along and make things ergonomically better, but also keep a manual part in there to maintain that flexibility and make sure that we have uptime. So we have found a mix of systems allows us to be more reliable and more flexible in our approach. Always looking at more automation, and we will continue to do so, but only when it fits the need of what we have to do to service our customers.
For our workflow analysis, as we previously discussed, we can run pre and post PSA simultaneously, allowing for quicker turnaround times. Also, in larger batches of our GC pesticide screen, we have multiple GC options, which allows us to split batches between two instruments to allow us faster runtime and turnaround time. We maintain multiple identical systems, which allows us to keep the workflow going if one of our instruments needs serviced. On to talking about our data handling. For our LC pesticide analysis portion, we use SciEx OS software for quantitation. Parallel processing is used for our large screens in order to make the workflow faster and run more smoothly. We accomplish this by dividing our large LC pesticide screen up alphabetically in order to perform the analysis. External calibration curves are used with the internal standard approach. We have created methods that will allow one chemist to analyze pesticides starting with letters A through I, and also analyze the pre-PSA cleanup portion of the analytes, while another chemist will analyze analytes J through Z. The chemist will then review each other's data to ensure the analytes are identified correctly. They meet ion ratio requirements, retention time requirements, and all other data acceptability requirements. After chromatography analysis and review, the data is all merged together on a spreadsheet and tolerances are checked for all sample detections as determined by Title 40, Chapter 1, Part 180 of the Code of Federal Regulations. Having multiple eyes on the same sets of data creates a situation in which systemic errors are more likely to be found. For our large GC pesticide screen, we use the Insight software from Shimadzu to analyze the data. Like the LC portion, we use external calibration with internal standard for the GC analysis as well. A chemist analyzes the data, establishes applicable tolerances for any detections. That data then gets passed along to another chemist who will review it to ensure all data acceptance criteria is met prior to reporting the data to the customer.
With the data handling, it's important to ensure transparency to clients regarding any performance impact to the data due to sample matrix issues. When we look at the data, we have data acceptance criteria and release criteria that may cause us to modify the QL, which would be a raise to the QL based off of if we can see a spike, but the recovery criteria are not acceptable. Not determinable results are reported when we know that the spike recovery is not working within a certain range, and not reportable results if the analysis is simply not compatible with that matrix.
Next, we'll go on to talking about the comparison with the FDA pesticide monitoring program. Here at NQAC, over 9,500 pesticide tests were performed between the calendar years of 2021 and 2022. As we discussed previously, we screened for over 400 pesticide residues in food, herbs, spices, and raw materials from domestic and foreign sources. A list was compiled of the pesticide residues detected as determined by Title 40, Chapter 1, Part 180 of the Code of Federal Regulations in different matrix groups for food samples submitted during that time frame. The U.S. Food and Drug Administration conducts a pesticide residue monitoring program and releases an annual report which is available on their website. The most recently published report is for data from the year 2020. They screened for approximately 750 different pesticides in both domestic and imported commodities. In the fiscal year of 2020, the FDA analyzed 2,078 human food samples, 316 domestic, and 1,762 import samples in its regulatory monitoring program. The FDA collected domestic human food samples from 35 states and import human food samples from 79 countries. In the comparison between the results obtained at NQAC versus the FDA monitoring program findings, NQAC detections align with the FDA findings, but were somewhat lower due to Nestle supplier management. In domestic samples, the FDA saw approximately 56% of samples with pesticide detections. In imported samples, the FDA saw approximately 40% with pesticide detections. The FDA uses a combination of MSMS and high-res detection, while NQAC uses only MSMS, as it's more definitive for meeting regulatory needs. As mentioned previously, our method scope is also designed for the screening in multiple markets, so the analyte focus may be somewhat different. Because of this, a direct comparison is not possible, but we generally see similar trends. One variable of note is that while the FDA is monitoring all products on the market, a significant portion of our testing comes through the Nestle supplier management process, meaning that the vendors that do not meet Nestle norms are systematically removed. As a result, our overall detection rate tends to be somewhat lower than what the FDA might find.
Evolving to meet analytical needs. As you can imagine, our portfolio needs to change every year to meet customer needs. There's always a new pesticide or threat out there that is considered, and it is our job to make sure that clients are aware and can monitor for those threats. Here at Nestle, we respond to new needs two ways. We have a top-down approach, which comes from Nestle Research. They monitor regulation and emerging threats and work to develop methods that meet those needs. And then a bottom-up approach here at the facility where we can develop or modify methods for the market need. Our pesticide method was designed with the current market of North and South America in mind, but that is also changing, and we will add compounds as needed, or sometimes analyses, as the case of polar pesticides, glyphosate, or ethylene oxide. Both NR, Nestle Research, and the NQAC engage in the scientific community to ensure that our analytical methodology is robust and recognized and up to date and aligned with the needs of the market. As a part of a global company and to meet the needs of our local customers, we have to make sure our pesticide methods meet both needs. For the local and US customers, we support them by providing guidance on the 40 CFR Part 180 that Angie mentioned earlier on those tolerances so that they can be apprised where their detections, if they have any, fall in regard to tolerances. The scope, again, has to both meet US and foreign market needs, and so we have designed the analyte scope to meet that. And the analyses for Nestle are required to meet European regulatory and guidance limits for infant formulas and foods. These are typically the strictest limits in the world, so it's important that our analyses can achieve these performance limits no matter what the regulatory limits are in a local region.
Well, thank you, Andrew and Angela, for your excellent presentation. We now have time for some questions. So our first question is, have you come across any matrices that have needed a matrix matched standard curve to accurately quantify? If so, have you found solutions to reduce the cost and work for those samples?
Yeah, that's I'll take that. This is Andrew Savage. It's a great question. I saw that question came in from an old colleague of mine from a while ago. So hello, I have to say. It's a good question. Matrix match curve is wonderful tool, and it's something that certainly would be wonderful to be able to use in an industry like this. That said, the types of matrices we get in on a daily basis really vary a ton. So we don't have that option per se. We have noted that we modified our GC method to use a matrix in the curve. We use a milk powder extract with it because we have issues with recovery on the GC if you use just straight up solvent. So no, we control everything based off of those internal standards, and the sample spikes are pretty much how we control it, and evaluate that then against that external solvent curve, which, like I said, on the GC side, has some matrix in it to help it perform the same as the extracts. I have certainly seen in some industries where you're running the same matrix over and over. That is a great way to help control that, but it's not something we could do here. So yeah, great question.
That's great. Thank you, Andrew. So our next question is, why do some pesticides get analyzed by LC and others by GC?
I can take that question. This is Angie. For that question, GC is suited to substances that are more gaseous under ambient conditions, or can actually undergo a change in state of matter to the gas phase with heat, such as volatile compounds. GC is limited to compounds that are low in molecular weight and stable at higher temperatures. Samples with non-volatile components or those prone to decomposition may not be suitable for the GC analysis and may need to be analyzed by LC. LC can handle volatile and non-volatile compounds as long as they are soluble in liquid phase. LC can also accommodate complex matrices and impurities in samples, and is a little more forgiving of the non-volatile impurities that you can see in a sample matrix.
That's great. Thank you. Another question we have that's come in is, how is it possible to detect all 200 pesticides in LC-MS with appropriate resolution?
Oh, that's also a good question. That is actually something with current technology available to most analytical laboratories, that with tandem mass spectrometry and appropriate liquid chromatography is not real hard to achieve. So the real advantage and power of mass spectrometry is you can deal with coelution as long as the compounds have individual or different parent masses, or even in the case of parent masses, different fragment masses. So this is something you can look through a lot of literature online from many of the vendors and find 200 is pretty easily achieved. The biggest limitation we usually would see on the number that we can look at is the scan rate of the instrument, with coupled with the chromatography. So with a 10-minute or so LC run and the speed that the instrument can scan through all the transitions, we feel pretty comfortable going up to about 600 compounds actually. So it can do that. The 6500, it's a very fast and very sensitive instrument. If you extended out the LC, you could gradient, you could even go a little further than that if you wanted. Great question.
That's great. Thank you. So our next question is, which spices and dried herbs have you found to be particularly complex matrices?
Oh yeah, this is Angie again. Yeah, we find that on the GC side, we have a lot of issues with the like spinach and kale, the that have the high chlorophyll content. We have issues with those, and then I know on the LT side we have a lot of issues with like black pepper, and some of the other matrices. They just produce interferences that mask the analytes that we're trying to analyze for.
Great, thank you. And we have our next question, which is, do you consider it necessary to evaluate sensitivity or suitability before starting routine tests?
Yeah, absolutely something that we do simply simply from a workflow standpoint. The last thing you want to do is put a sequence on the instrument and then go home for the day, and then come in the next morning and realize there was an issue with the instrument. So our workflow here, typically while the samples are being prepped, a chemist will set up the instrument with the initial calibration curves and blanks and any other controls, so that we can see that the instrument is sensitive and stable. If there's an issue, say the auto sampler is not picking up the sample reproducibly. When you run a calibration curve, you're going to note that right away. It's gonna be all over the place. Sensitivity wise, of course, as well. One of the first things you're going to do after you start up a sequence and have the instrument equilibrate is look at that low standard and make sure you have plenty of signal noise in all the transitions. So yeah, good question.
That's great. Thank you, Andrew. So our next question is, are all of your methods targeted, or do you also perform untargeted analysis?
Yeah, I'll take that one. So that's a good question. And it's something that if you go to different food safety conferences, you're going to see a lot of presentations with analyses done both ways. Currently, we only do targeted analyses. Untargeted analyses are quite interesting in what you can determine in that. But our goal here is to give our customers actionable information on what they need to do with that food. So untargeted analyses, while I do think there's a lot of future in that, there's some challenges with making, if you detect something, do you have a reference material to truly quantitate it right there? If you detect something and it's not a regulated compound, what are you going to do with that information? So untargeted analysis is a really good way to determine, in my opinion, what you need to do targeted analysis for. Targeted analysis is typically going to be it's a little easier to build into a robust workflow as well, and you can usually be a little more sensitive on that as well. So what I've seen is often the university and research side, or the regulatory side, they'll start using untargeted analysis to determine what they need to be looking for and concerned about. And then on the industry side, we're going to be using more of a targeted approach to make sure we're meeting those requirements. So, thank you.
That's great. Thank you both for answering those questions. And if any other questions do come in, I'm sure that Andrew and Angela will be happy to respond to them via email. Thank you also to our scientific partners in this webinar, SciEx. And with that, I'd like to thank you all for logging in and listening. Thank you and goodbye.